Good morning, class. So for chapter 18, I definitely wanted to help guide us a little bit here. Good morning, class. So chapter 18 has a lot going on, and I was actually reflecting on the fact that this is probably the stuff that has changed the most since I was in college, since I took this class. So chapter 18 greatly expanded over the last 20 years, and I want us to kind of narrow things down in this chapter. So I want to make it really clear what I expect you to know and what is kind of interesting, and by all means feel free to delve into it, right, but might be more for your own interest. So when we talk about gene expression, there's multiple ways to say this. So chapter 17 was about transcription and translation. That is gene expression, right? Taking the gene and actually turning it into the protein, right? That's the expression. That's what we're going to see. So this chapter then is a lot more about how we turn transcription translation on and off, right? Or how we modify it in certain circumstances. And this actually ends up being really important, particularly in multicellular organisms. What we'll see is this is what is going to let cells be specialized, right? Every cell in your body has the exact same DNA, but the pancreas, in fact, the beta cells in the pancreas are the only cells in the body that express the gene for insulin. Every cell has the gene, so why is it just the pancreas, right? So we would call that differentiation, that different cells in the body have different roles. So this idea of specializing, right? Particularly in a multicellular organism, that's pretty cool that different parts of the body do different things. We'll also see that this regulation of gene expression is super important, particularly maybe in bacteria, for energy savings, right? If you think about a unicellular organism, it's completely exposed to the environment all the time. That environment is changing. It doesn't have homeostasis to relax in. And so it is going to deal with that changing environment by turning on and off certain genes. It only wants to, say, make a certain protein when it has to, right? So energy or nutrient savings. And so the other way to think about this idea of regulating gene expression, this again, biology is just changing so rapidly. There's a whole nother field of biology, right? We had genetics, that's been around since what, the 40s or 50s, right? We watched in the gene. Now we have a field called epigenetics. And epigenetics is basically anything outside that little coding sequence of the gene that actually affects the expression of these genes. So kind of what we learned is, we get really excited when we scientists discovered the whole genome of a human, right? We mapped the human genome, and then we realized, it's actually much more important that the cells know like what to do with that information, when to turn it on and off. And so epigenetics looks at this whole other field that says, why do we express some genes and not others at certain times and not others? So let's take a look. We're going to start out, let's start here. So a metabolic pathway. A metabolic pathway is just when you have a series of enzymes that basically have to work in an order to produce something. So in this particular example, and we will see this one again throughout this presentation, right? You have some molecule, and they just made it in orange box. They don't want to bog you down in details right now. And we have an enzyme that converts it a little bit. Now it looks like a coffin. And then enzyme 2 comes in, right? And it's starting to look a little more like a molecule maybe that we're familiar with. There's enzyme 3, and here's our final product. Now this final product in this particular example happens to be tryptophan. Tryptophan is one of our 20 amino acids. You might recognize that from earlier chapters. So here is the key thing, and you will see this over and over if you go on and take anatomy with me, this idea of negative feedback. So notice once we've made tryptophan, we've got red arrows going both ways here with a little minus sign. So the idea here is we have two ways to kind of say, hey, we've got enough tryptophan. right? And that's what negative feedback is. Negative feedback says, hey, the end result of a reaction, or in this case, a series of reactions, is to negate that reaction. So once we have enough tryptophan, we want to turn off this metabolic pathway, right? We've got enough. Let's save our time, our energy, our enzymes, etc. So one example of negative feedback is what we're calling feedback inhibition. This would be when the tryptophan actually binds to this enzyme, making it so that it doesn't function properly. And that's actually really common, that the end result of a metabolic pathway would actually bind early in that pathway and shut it off. Okay, what this chapter focuses more on is another means of negative feedback, and that is the regulation of gene expression, where tryptophan, the end product of this metabolic pathway, these 3 different enzymatic reactions, actually turns off the genes that code for these enzymes, right? These enzymes didn't come from nowhere. They're proteins. They were produced by transcription and translation. And so now we're going to use tryptophan to actually turn off transcription of those genes. Don't even make these enzymes so that we're not tempted, say, to make more tryptophan. Okay, but both of these are negative feedback. They're great things. Negative does not have any kind of moral connotation. Negative just means, right, something was on, we're turning it off. Something was off, we're turning it on. That's super important in this idea of homeostasis, right? How do we hit a set point? Well, if you're getting too warm, you sweat and cool off. If you're getting too cool, you shiver and heat up, right? It's the same idea. Okay, so a little bit of homeostasis here. Okay, so we are going to start by looking at this in bacteria. Bacteria is where this was first understood. It's in many ways much simpler. Remember, bacteria have a single chromosome, way fewer genes. But I will emphasize, this is not how you or other eukaryotes regulate your genes, right? This is only going to be what bacteria do. So in particular here, we're looking at E. coli. Now remember, in bacteria, single-celled organisms constantly exposed to this changing environment. There's not really homeostasis. And so the enzymes and the proteins that they're producing are constantly going to be changing. So what researchers realized was that when there's a metabolic pathway in a bacteria that requires multiple enzymes, all those genes are packed together in the chromosome, right? So in this particular example, this again is, we're going to use a kind of more in depth, that first example of making tryptophan, right? So here they're showing us we actually need, right, these different components of enzymes, right? So we need to build these proteins, E, D, C, B, and A, and notice their genes are all together in the chromosome. Eukaryotes apparently aren't this smart. We stick, the genes for eye color are on 3 separate chromosomes, right? That sort of thing. But in bacteria, they're metabolic pathways, enzymes, have all their genes tucked together. And so what researchers did is they called this an operon, right? It's a play on words. Opera means great work. And they decided, wow, the bacteria, they've got this pretty organized. This is pretty slick. Okay, so let me show you this. What an operon has is 3 components. It has a promoter, an operator, and then the actual genes, the structural genes, right? So those 3 things make up the operon. Now, the promoter you might recognize as the place where RNA polymerase docks, right? And RNA polymerase wants to run along this gene and do transcription. The operator decides if that happens or not. Okay, so if the operator, see how there's like 3 little docking stations here? If a repressor molecule, like this guy down here, is inactive, it can't bind, right? See how the inactive repressor is kind of curved. It wouldn't fit up in those docking stations. So when that operator is unbound and open, sure enough, RNA polymerase can run through here and do transcription, right? Make its messenger RNA and then translation makes the protein. Now, that's what's going to happen when tryptophan is basically absent, right? The repressor is then inactive and the operon is in the on position. Okay, if tryptophan is present, right, we used these enzymes and we made a bunch of the amino acid tryptophan. Oop, there's tryptophan. See our little amino acid down here? Yep. Tryptophan fits into that repressor and activates it, right? Notice it actually changes shape so it fits into the operator. Now the operator is blocked, and look at poor RNA polymerase, right? He can't actually dock. He can't get onto the messenger RNA like he wanted to. There's no messenger RNA made, right? So transcription doesn't take place. So we've turned this operon off. Now, a couple things to keep in mind here. If we're building something, if we're building tryptophan or anything else, that is considered an anabolic pathway, a building of a more complex molecule, right? You take anabolic steroids if you want to build muscle mass, right? Anabolic reactions are building up, building something complicated. And so anabolic pathways are going to use a type of operon that's considered repressible. So the way I think, right, to repress something means to like stop it. So the way I think about this is in an anabolic pathway, right, our E. coli here, it needs tryptophan. And so kind of the status quo here is that operon is on and we're building until we have enough tryptophan, or I don't know, maybe it's Thanksgiving and you land on a turkey and there's tons of the amino acid tryptophan. You don't want to waste your nutrients. You don't want to waste your energy, right? And so once there's plenty of tryptophan, you can turn it off, right? You can repress the operon. So anabolic pathways use repressible operons. So, okay, on one hand, you look at this and you're like, holy cow, that's a lot of details. But on the other hand, isn't this slick? I mean, there's a reason they called this the operon, the great work, because bacteria have this figured out, right? They're being extremely efficient here. Okay, so let's look at the flip side. Let's look at what happens. Oh, first, we're zooming in, right? So again, this is when the operon is on and we're making our enzymes to make tryptophan. Here's when the operon is off because we have plenty of tryptophan activating our repressor. Okay, now we'll look at the flip side. So then what happens in a catabolic situation, right? So there's actually lots of catabolic pathways as well. I can do this, catabolic. And catabolic pathways, that's a B, sorry. And catabolic pathways are the opposite. They're taking complex molecules and ripping them apart. I just picture like a cat ripping things apart. Okay, so the classic example in E. coli is the lac operon. This is the lactose operon. So if you recall, and sadly they don't give it to us, lactose, that's terrible attempt at drawing here, right? Lactose is a disaccharide, right? It's two monosaccharides, two simple sugars hooked together. Our bacteria really just wants one of them. That's the glucose. And so we need to be able to rip lactose apart. But lactose isn't particularly common in the environment. So what our bacteria do in a catabolic pathway, they use what's called an inducible operon. Right? What does it mean to be induced? Maybe you've heard that term for pregnancy, for delivery, right? You get induced to like turn labor on, right? To start the process. And so that means to start off, you're not in labor, right? This operon isn't functional. So let's take a peek at this, right? That's exactly what happens. In normal situations, there's not a lot of lactose around, right? So lactose is absent and the operon is off. Right? What makes an operon off? It's when the repressor is active, it's bound at the operator, and RNA polymerase can't run transcription. Okay? But say it's Thanksgiving, right? We had the turkey last time. Now you've got a pile of Cool Whip, and this bacteria lands in the Cool Whip, and it's like there's lactose everywhere, so much sugar, right? Sorry. So when there is lactose present, and they're showing lactose here as this little green sphere, lactose binds to the repressor and inactivates it. See how its shape changed a little bit, and it no longer fits. So now RNA polymerase can do its thing, right? It can run along the gene and make the enzymes necessary to break lactose apart. Isn't that slick? So we turned it on, we induced it when that substrate was present. So again, zoomed in here, right? When the repressor is active, no transcription takes place. When lactose is here, it inactivates the repressor, that opens up the operator. RNA polymerase can do its job. Okay, it gets more complicated than that, but that's all the further I want us to go. That's plenty, right? Again, thinking about anabolic pathways using repressible operons, what did I just say? Yep, catabolic pathways using inducible operons. Okay, you should definitely know those three sections of an operon and this kind of general, I'm totally in the way here. This general outline, again, talking about inducible, I'm on the wrong screen, sorry. Inducible, and catabolic, repressible, and anabolic. If it's any incentive for learning this now, I literally just went through the same thing in microbiology, except the chapter in microbiology has DNA replication, transcription, translation, and gene regulation all in one chapter. So this is your chance. I know it feels like it's going fast, but this is your chance to actually get some of this down. Okay, so we are going to switch gears to eukaryotes. And this is where I really want to rein the chapter in, right? Eukaryotes, the gene regulation in eukaryotes is much more complicated, right? There's more DNA. We have this idea that we need to express different genes in different cells. And so there is a ton going on. And so what I want us to focus on, there's two images that I think do a really nice job of summarizing it, right? So this is figure 18.6. Okay, and it's running through, we should recognize some of these processes, and then we're just going to pull out a few things here, right? So here's our DNA. This screen. Here's our DNA, and notice there's transcription, right? So in transcription, we take the DNA and we make RNA. But what we're going to find is there's a level of gene expression that can get altered at transcription. I should start before that. Okay, back up. In order for the DNA to be accessible to get transcription to take place, we have to kind of unpack it out of the chromatin. Okay, so we'll look at how that happens. We'll look at the fact that during transcription, we can decide what genes to transcribe and what not to. We'll talk about some of the things that happen in RNA processing and how that might lead to differential gene expression. Once that messenger RNA enters the cytoplasm, right, there are enzymes there that are trying to degrade it. How quickly it's degraded can determine if it's expressed or not, right? Translation taking place at the ribosome has some things that we can control. And then finally, after translation, so some people call it post-translational control, we've got a few things that we can do with that polypeptide, right? We have to process it, fold it into the appropriate protein. There again are molecules that are trying to break this protein down, and or we have to make sure that it gets to where it's actually going. So if you want to say get that protein out of the cell, it might have to go through the ER and the Golgi apparatus, which we'll talk about, right? So there's a lot more going on here, and the book does go into great detail. I just want us to have it kind of at this level, Okay, and so this is figure, what, 18 UN 10. I don't know what the UN is for. And so notice it follows that same pattern, right? Chromatin modification, where's my chromatin modification, transcription. Here's that RNA processing, RNA degradation, translation, and that protein processing and degradation. You should know that those steps exist. And then sorry, I'm in the way there. Okay, protein processing and degradation. We are going to look at each of these a little, but you should realize, one, it's more complicated, and that these are kind of the 5, 6-ish areas where control can happen. Okay, so first one, this idea of chromatin modification. So remember that DNA is wrapped. So DNA is in this blue strand here, the double helix, is wrapped around proteins called histones. Wow, in the way again. Right? And this is used to package these huge long strands of DNA, right? Billions of base pairs long, into this microscopic tiny nucleus. You wrap it up on tiny proteins and condense it into chromatin, right? But what we find is that when the DNA is tightly packed, we actually call that heterochromatin, and it's basically inaccessible, right? So notice here, the compact DNA, let me get my marking pen back. Compact DNA, when it's wrapped tightly around those histones, when it is heterochromatin, it's not expressed. Okay, we actually have to do some unwrapping in order to have, see now there's like a little section of DNA here that we could actually get that molecular machinery around. So this is a level of gene expression control, right? Is it tightly wrapped as heterochromatin? Is it more relaxed as euchromatin? And what we'll find is you have a series of enzymes that kind of help move where those open sections are and determine what gets expressed. Again, I kind of think of this as like, if you happen to remember VHS, right? You have like tape wound around. It's only around like these little spools. I think of those spools as the proteins, right? And then the tape with all the information is the DNA. And it's only that little segment that's actually open, right, to the reader that's being expressed or showing up on your TV. Whatever's wrapped around the spool at the time is not what you're seeing. You might see it later, right? So we can move this and change the gene expression. Okay, that's it, right, for chromatin modification. And again, they do a nice job in this image of kind of spelling that out for you. Genes in highly compacted chromatin are not transcribed. They're telling you more specifically the process is called acetylation. Don't worry about it, but realize when it's more open, that's when we see transcription taking place. Ooh, last point, I will mention this. This idea of methylation. Methylation is the addition of a methyl group, a CH3, right? Methane is CH4. This methyl group, this has been a hot topic for research recently on epigenetics, on what turns genes on and off. There's some really fascinating studies. Things like stress can change where we put methyl groups and actually change gene expression. And what gets even weirder is that if you're super stressed and you put all these methyl groups onto DNA, you can actually pass that on to offspring. Anyway, super crazy, really interesting research there. So you might want to check that out at some point, maybe when the semester's over and you have free time. Okay, so that's chromatin modification. Then we move on to transcription, right? And so transcription has a couple things going on as well. So again, we talked about this idea that there's a promoter region, right? Recognize that ta-ta box, the TATA, that kind of signals RNA polymerase to come in. But notice it's actually a little more complicated than that. So you have what are called transcription factors. And so it's not just like sorry, I kind of feel like I lied now last week. It's not like RNA polymerase just shows up. He's like, oh, promoter, here we go, right? There's actually a lot of these transcription factors, a variety of different proteins that actually set up this whole situation, right? The DNA actually folds over on itself. And then when everything is present, RNA polymerase is actually going to pop in and go ahead and do RNA synthesis. Okay, so a little more complicated than maybe we let on in the last chapter, which seemed complicated enough as it was, right? And again, this is trying to show the same kind of idea here, right? So they're showing you two different genes, the albumin gene versus this crystalline gene. And notice you have slightly different enhancer regions. So you set up a different series of factors, and that determines what cell is producing albumin, right? Polymerase is working here, versus whether crystalline is expressed, right? And that is going to make it so this cell producing crystalline, right, that is only happening in the lens of your eye, and the liver cell is the one producing albumin. Right? And so different sets of transcription factors in different cells helps us have those different roles. Okay. So again, here's your summary looking at what's going on with transcription. So then we move on into processing, and we already talked about that a little bit more when we talked about transcription translation, okay, because we do have this ability to alternatively splice those pre-mRNA or messenger RNA transcripts, right? And so it looked something like this. Get out of your way, okay? Remember, exons and introns. So exons are going to be expressed. Wow. Exons are going to be expressed. And so what we're doing here is we're pulling these introns out, right? We're splicing together, fusing together all of the exons. So there's our dark orange. And now between a start codon and a stop codon, right, we have the actual messenger RNA that we want to make a copy of. Now, where this turns into a level of gene expression control is that you can actually run alternative splicing. Okay, so, and how do I want to say this? An intron isn't always an intron, right? And so notice we did a little differential splicing here, right? In this particular side, we get 1, 2, 3, 5. On this side, we would get 1, 2, 4, 5. Different RNA codes, those are actually going to make slightly different proteins, okay? So how you splice the DNA is going to make a difference in how that gene is actually expressed. Okay, so alternative RNA splicing. That one's maybe the easiest. Okay, so then we start getting down into translation. And where's my little pen? There we go. We get into translation. And again, this is happening at the ribosome. The only thing I would say about this level of gene expression control is that the initiation, the starting of translation, can be controlled. So just because a ribosome grabs something doesn't mean it's always going to transcribe it. But what I want to focus on a little bit more are these ideas of protein processing and degradation and what happens to the messenger RNA as well. So if translation just happened, okay, we're not done, right? Translation technically gives you a polypeptide chain. And so we need to make sure that folds properly, right? And there's actually chaperone proteins that do that. Then this protein doesn't necessarily get to live forever in the cell. And so what they're showing here, this is the protein that we've decided we want to get rid of, right? We made enough of it. Or if you think about something like Alzheimer's disease, the problem in Alzheimer's is there's these protein plaques that start building up on neurons. Maybe those proteins should have been degraded. We needed to use them, but then we should have gotten rid of them. And the way cells do this is they use this little tool called a proteasome. And so you kind of like shove your protein into this proteasome, and it goes ahead and fragments it, right? So you have enzymes that are going to help break these proteins down. Could it be in something like Huntington's and Alzheimer's, where it's actually an excess of protein that's the problem? Could it be a problem with proteasomes? Interesting. So again, that's going to alter this expression, right? The overall amount of protein in the cell makes a difference. We also regulate the number of RNA transcripts that we leave sitting around in the cell. So again, we talked about how the cap and the tail on messenger RNA is keeping enzymes from breaking it down. We'll also find that we use, this is crazy, a little molecule called a microRNA. So it is RNA, it's coded for in your genome, and it requires transcription to make these microRNAs. They're called microRNAs because they're tiny, right? Notice what it does. It finds a target messenger RNA, right? It has to bind to it, okay? And then it can actually degrade it, right? It breaks that messenger RNA down, says, hey, we're done. And students often find this frustrating. We're like, well, why did we make it if we're just going to break it down? But it's this idea that it's useful to turn genes on, but you also need to be able to turn them off. If this messenger RNA hung out forever, and so you just kept making and making and making insulin, you lose control over blood sugar. And gene expression is really about controlling kind of those protein products that we wanted to make, right? And so again, I think this particular image is really nice at summarizing. So feel free to look into any of these areas further, but do not get lost in the weeds. There is a lot going on here, and some of it's super fascinating, but we're going to keep it a little bit tighter for general college biology purposes. Okay, couple last things in this chapter that I want to mention, again, without going into great detail. So look through these sections. The dynamic study module will take you through here. Okay, but we're going to look at this actually fairly quickly. So again, when you're thinking about gene expression, one of the things to realize is that a lot of this becomes super important in embryonic development, right? You start out as a fertilized egg, a zygote. Where's my next? Right? You start out as a fertilized, oh, there's the sperm, there's the egg, a fertilized egg, also called a zygote. Okay, that brought the two gametes, the two products of meiosis together to get back to a diploid cell. And now we're going to start doing cell division, mitosis, to get 2 cells and then 4 cells and then 8 cells. So notice one of the coolest things, right? But so it's like, how do you go from a fertilized egg to like even this tadpole, like knowing where eyes go, where a tail goes? There's a lot of research in this kind of showing that some of the factors, notice in this egg that down this end of the cell, there's lots of spheres. And on this end of the cell, there's lots of triangles. And so even like how things are arranged in the cytoplasm, as this cell starts to divide and divide and divide, those molecules can actually act to turn on and off certain genes. And so that's what they're trying to show down here, right, is that those particular, like the orange spheres here, are affecting the nucleus and gene expression. I just think that's phenomenal. So again, a whole field kind of this developmental biology looking at how gene expression changes during development to make sure you end up with eyes on one end of the body and a tail on the other. Okay, again, keep it fairly superficial there. We also run into this situation where cells need to differentiate. So again, in this case, they're trying to show that we are wanting to become muscle here, right? So how do you go from a stem cell to that actual differentiated cell? And so they call this determination, when you irreversibly commit a cell to becoming a particular cell type. So you're no longer a stem cell, you can't divide and become whatever you want. And so there are genes, right? Here's a master regulatory gene saying, okay, now this particular cell, once this master regulatory gene is turned on, then we can express all these other genes that we need to be a muscle cell. There are genes, again, here's your fruit fly, still a common research subject, genes that have been identified that help the body lay out a pattern, right? So like, where should the head be? What are the different body segments? Because again, this happens the same every time you have a fruit fly hatching out of an egg, right? Again, the mutants often help us understand more about this. So here, this is the craziest thing, right? Mess with some gene expression and you get legs instead of antenna, right? The antenna don't have to be there. It's just because a set of genes are expressed, right? If you switch that gene expression, you can put a leg there. There's no reason you can't. Now, of course, this wouldn't be useful for a fruit fly. This wouldn't survive. And that is true of many mutants, right? A lot of times mutations are lethal, but they often help us understand what the correct gene is supposed to do. Again, genes identified. This one's called the bicoid gene. This is the gene that actually tells like the front half of the body where it's going to be. And so this picture is just trying to show, right, more of that messenger RNA of the bicoid gene in the front end, right? And that is going to determine the gene expression of those cells. Okay, last thing then. Speaking of mutations, humans are often interested in mutations because in humans, mutations often lead to cancer. Not always, there's all sorts of other, obviously, genetic issues that we were looking at. But what I did want to point out here is that really these mutations fall into two camps. Mutations that are going to lead to cancer fall into two camps. Either we are altering a gene so that we're making too much of a protein, right? And that could lead to increased cell division. When it comes down to it, cancer is a result of a DNA mutation, and that mutation is making it so cells are dividing out of control. So you have genes that control protein production, right? That if you mutated it, you could stimulate cell division. And you also have a set of genes that make it, that should stop division. But if you don't produce the protein that should stop division, again, that's going to allow this overproduction. And so these genes that normally drive replication, right? We should have some of them expressed and tell cells to divide when needed. These are called proto-oncogenes, and they mutate into oncogenes, into cancer genes, right? And so again, without getting into too much of the detail, right, we should have normal growth stimulating protein. Whoop, that's still showing excess. Oh yeah, we've already mutated into an oncogene. The proto-oncogene is normal, right? We should have some of that protein expressed. Oh, they're trying to show some of these mutations. So you could get a new promoter, which would make you overexpress the protein. You could make too many copies of that gene, and now you're making too much protein, etc. The point here is oncogenes are overexpressing and overdriving cell division. Okay, and so this is what it would look like. And this is another one, where there's a growth factor that should be binding on the outside of the cell. Again, without getting too many of the details here, right? This growth factor is binding on a cellular receptor and should be triggering transcription, right? We get a normal amount of protein, a normal amount of cell division. So you need this, right? Like if you scratch your skin off, you want these growth factors to tell cells to divide and replace themselves. But you might get a mutation, right, where it doesn't matter, right? Notice that growth factor isn't bound here and we're still doing transcription. And so you get an overproduction of that protein. Okay. This is an example of where our stop sign isn't working. So this set of genes that can also be mutated are called tumor suppressor genes. So they should be stopping cell division. So notice what happens here, right? As long as the tumor suppressor gene is correct, it's producing this protein. It's called P54. We're going to get transcription of our stop signal, right? And we don't get cell division. And this is actually really appropriate, particularly if there's DNA damage. But if there's a mutation in that tumor suppressor gene, right, and we have a mutated P53, right, now there may be damage to the DNA and we don't stop the cell from dividing, right? And so again, both of those are going to contribute to cancer. We kind of get uncontrolled cell growth, like the gas pedal on your car is stuck to the floor, right? We're dividing, and the brakes went out, right? There should be a normal protection by tumor suppressor genes. Okay, I gotta run to, sorry, I had to run to lab, but really just wrapping up. The thought here with cancer then, right? Again, all cancers start with a mutation of the DNA. And particularly this is taking place in genes that regulate the cell cycle, that regulate when cells are going to divide. So again, proto-oncogenes, if they mutate into oncogenes, the cancer, the cells just divide out of control. The tumor suppressor gene, if that gets mutated, it's not going to stop the growth. And so this ends up leading to what some people call like a two-hit hypothesis, or here they're talking about this multi-step model. And the idea is that cancer doesn't come from one mutation. So notice what's happening. In this case, they're looking at colorectal cancer. And so you have a loss of a tumor suppressor gene and you start getting like a small polyp. And then here, notice this oncogene, right? So we had a mutation going from a proto-oncogene to an oncogene. So that gets turned on and we're losing another tumor suppressor gene, right? And then it's getting bigger and then we lose another tumor suppressor gene. And really this P53 is a major, is implicated in a lot of different cancers. That's a major tumor suppressor gene. So then by the time you get, what is this, like 4 different mutations going, now you have this malignant tumor, right? And when we say malignant, now we're actually talking about cancer. It's able to metastasize, it's able to spread to other tissues. So again, some of that comes back to gene expression, gene mutation as well. Okay, I think that's it. We'll catch you to talk more about this next class.